Right. This page I'm just going to really skip over. If you read your book in chapter 17, it talks about the 3 different types of RNA polymerases and what their specific jobs are. And if you start to hear all the things RNA polymerase, it sounds way too good to be true, and you're right. RNA polymerase doesn't do it all. Different versions of RNA polymerase do. Okay? RNA polymerase 2 is the one that transcribes the genes into mRNA, and that's the primary one that we discuss here in bio. This promoter sequence is important. The promoter sequence, again, is this TAT box, if you look here, TATA box. And this is the binding site. Now, eukaryotic cells have to have things called transcription factors, which is a bunch of proteins that are going to bind at this TAT site and get it ready for RNA polymerase to come in and start performing transcription. This differs from eukaryotic cells that don't need all this big introduction. So here is the initiation process in eukaryotic cells. And you have the promoter region, that TATA box. And what ends up happening is you have a bunch of proteins, which we call transcription factors, binding to this promoter region. You must have the transcription factors in order for RNA polymerase to begin transcription in eukaryotic cells. Prokaryotic cells do not require the transcription factors to bind before it actually starts doing its job. So transcription in eukaryotes cannot begin until transcription factors bind to the promoter region. That's an important detail, and it's got a slide all to itself. So make sure you understand, not only can regurgitate this for the test, but you understand it. This is showing you a promoter region, and these little guys right here are what we would call the transcription factors, or the proteins that make transcription occur, so that then this blue guy right here, RNA polymerase, can come in and start doing its job. Now, as I alluded to earlier, prokaryotes, RNA polymerase does not need transcription factors to actually mediate the binding of DNA to that initiation site. It will, prokaryotes are just able to start out. And this is a great image here. I like this image because you see, here's a prokaryotic cell. And this is the promoter region, and RNA polymerase can just get going. Here is a eukaryotic cell. And if you look, all of this is going to be your transcription factors in that promoter region. So all this stuff right here. And this is your RNA polymerase coming down right here, okay? So this just shows you that there is a lot of transcription factors that are required for eukaryotic cells. Here's another question, which again I would suggest to pause, answer, and then come back. Transcription in eukaryotes requires which of the following in addition to RNA polymerase? The answer is D, several transcription factors. D. What is the enzyme responsible for unwinding the double helix during transcription? This question's easy, but so many of you get it wrong because it's too easy. You see unwinding and you immediately go to helix. Helix still does the unwinding for RNA, but you cannot put it down as the reason it goes, as the reason it happens. All right, when you match RNA bases with DNA, remember this is just reminding you that when you would use a T, like here's an A, so A usually goes with T, you use a U for RNA. And if you look, this is another one of those pictures that you really suddenly see a big detail that you need to understand. What end is RNA polymerase adding to on this RNA strand? It's the 3' end. Just take note of that.